So I'm currently living in Dallas, Texas, and today is the first somewhat cold day in November. And it got me thinking, which is something I don't do too often, so when it happens, I take notice. Now, we know that psychrophiles are cold-loving organisms that like to put roots down in glaciers, mountain peaks, permafrost, places where most life wouldn't dare to exist. And it's not just the cold they have to contend with. Many of these individuals who live in the deep ocean have to deal with things like major pressure changes. So my question today is two-part. First of all, who the hell are they? And second of all, how in the world do they manage to do this? Like, what is it exactly in their toolkit that allows them to survive? Answering the first part of my question was pretty simple. To begin, we have our microscopic celebrities like archaea and bacteria, these sort of super simple single-celled organisms that are just everywhere all the time. They're insanely adaptable. But we also have some other guys hanging out like fungi and algae, and we even have some microscopic invertebrates like tardigrades. You might have seen them in the movie Ant-Man. They look like little bears. They're super cute. Answering the second part of my question is a little more tricky. When trying to break down the psychrophile toolkit, I think we should start with what I like to refer to as enzyme acrobatics. See, just like any other biological organism, at the heart of psychrophiles biology is their cells, and more importantly, the biochemical reactions that take place in and around those cells. And if we've learned anything from biochemistry, it's that enzymes are damn important. We want to understand how their enzymes manage to function in the cold when most other enzymes would just freeze up. And ultimately, this comes down to flexibility. The hard work of evolution has allowed these psychrophiles to enjoy the disappearance of various stabilizing interactions, resulting in more flexibility of the enzyme conformation. And fun fact, it's actually this trait that has made their use in modern-day detergents so important, by allowing us to wash things at lower temperatures without compromising on cleanliness. While this is a superpower in the cold, it makes them hypersensitive to heat. So if you warm them up too much, the enzyme will just unravel and it won't be useful anymore. It's like they've traded out their tough armor for a light jacket, so it makes them more flexible, but it won't give them any protection in a heat wave. Next in their toolkit, they have two very unique proteins, with the first acting as a cellular antifreeze, which helps to answer the question of how do they keep themselves from freezing solid. These unique molecules, called antifreeze proteins, or AFPs for short, basically act as little molecular snowplows, clearing the way and preventing ice formation from wreaking havoc inside the cell. And then you have sort of the AFP brother, which are ice nucleating proteins, or INPs for short. And instead of preventing ice formation, instead they act as ice architects. They control when, where, and how ice forms inside and around the cell. They control the blizzard. Some might even call him a blizzard wizard.
You shall not pass!
I don't know. The final tool these suckers have are compatible solutes. These compounds help psychrophiles adapt to the cold by lowering their cytoplasm's freezing temperature. Now, they're kind of the unsung heroes of cellular survival in the cold because they're not quite as flashy, but nevertheless very important. See, when water freezes outside the cell, it can actually draw water out from inside the cell, causing it to shrink and potentially die. Water wants to rush across that membrane to rectify the imbalance, but compatible solutes ensure that that osmotic balance stays put and prevents the cell from shrinking up like a raisin. There's also evidence to show that compatible solutes act as a built-in humidifier in the cell, so they're not just preventing freezing and shrinking, but they're also maintaining the cell's overall internal environment. Now, I could have gone much more in depth, and maybe I will in the future, but I think that's all I got on psychrophiles today. This seems to be a pretty good starting point. If you guys found value in this video and you like this type of content, please feel free to like and subscribe, and I'll see y'all next time. Peace.